These are the genes of the lac operon. Genes are a distinct sequence of nucleotides and are the basic physical and functional unit of heredity. This is RNA polymerase, and the role of that is in protein synthesis. It reads the genetic sequence in the DNA and transcribes the sequence into an RNA sequence. The RNA sequence will then be used in transcription. The role of ribosomes in this process is to read the genetic code in the mRNA and translate it into a sequence of amino acids that grow into long chains called polypeptides. The polypeptides will fold into proteins. They do this by attaching their large and small subunits around the mRNA strand and attracting the corresponding tRNA molecules that have amino acids attached. This is the operator. The role of this in the DNA sequence is the site for a repressor protein. This controls whether the transcription of a gene can occur. A promoter is the binding space for the RNA polymerase that marks the starting point for transcription. Structural genes code for the enzymes or other proteins that are required for specific biological processes. The presence of lactose will act as an inducer. When lactose binds to the repressor, it takes it away from the lac operon, allowing DNA polymerase to go through and code for the genes. In the absence of lactose, the repressor stays on and DNA polymerase is blocked by the repressor and cannot move and code for the genes. The regulatory gene is what controls the operon. It produces a small protein that's called a repressor that prevents the gene from being turned on. When the lac operon is activated, enzymes are produced to break down the lactose present. This lactose will be broken down into glucose and galactose. Then the glucose will be used for energy. When the lac operon isn't inactivated, no enzymes are produced to break down lactose because it isn't present. The RNA polymerase is unable to read and transcribe the gene because of the repressor being present. The inducer in the lac operon is allolactose, which helps turn the gene on. A mutation in the regulatory gene may cause the gene to be permanently turned off or on. A loss of the promoter site would cause the RNA polymerase inability to attach, so no enzymes would be produced. The function of an operon is similar to feedback inhibition because feedback doesn't allow a cell to waste energy in substrates. The operon is turned on and off if it's needed to function and produce proteins.